So I'm Ricky Rickles. I'm a Senior Director of Oncology here at Horizon Discovery at our Boston site. A little bit of background about myself. I moved up to Boston in 1989 to work at Harvard University Biochemistry Department with Mark Potashny. I got an offer I couldn't refuse, which was to join Ariad Pharmaceuticals when it first started, and I worked there for about 7 or 8 years with Joan Brugge, and we helped to start the Herx Area Genomics Center that became the Aventis Genomics Center. I was head of the genetic screens group, a sector head, and then I went on to work at Vertex Pharmaceuticals for 2 years, and then I left industry after a total of 11 years to go back into academia to work with Steve Elledge at Harvard Medical School on this new enabling technology around gene silencing with shRNA and viral vector systems. Spent 2 years there, and then I got an offer I couldn't refuse, which was to join at that point a company called Combinatorics that was focused on development of a platform that I'll discuss with you today to do combination high throughput screening. And so overall, I've been here at this site initially as Combinatorics, and we're lucky enough a year ago to be acquired by Horizon Discovery to be now part of Horizon with their great enabling technologies that I'll discuss with you and moving forward with this technology and a number of particularly interesting ways that we hope will ultimately not only help provide value to folks doing research on drug discovery, but most important, to increase the efficacy for the various therapeutics that are being developed, not only for oncology, but also for a number of other therapeutic areas. So with that long-winded introduction, let me just give you now a brief overview of Horizon Discovery, which essentially initially started off as a company that was doing very exquisite gene editing of mammalian cells, initially with recombinant AAV, and then it's clearly kept up with the times so that we're now one of the leaders in the field with respect to CRISPR-Cas9 technologies for gene engineering. What's been really great is the explosion of information that's become available through next generation sequencing, such that we can now incorporate that information into our thinking about the animal models and cell-based models that we work with to generate appropriate cell lines, not through use of expression vectors or knockout, but we can actually knock in specific mutations into cell lines, for example, or essentially recapitulate any sort of variation we see in the human genome into our cell lines to test the impact, the functional impact in doing so, which can help in terms of target ID as well as target validation, but particularly important since we also do both in vitro and in vivo screening, we can move forward with respect to assessment of various agents as potential therapeutics. We also, as a result of the isogenic cell line generation, we have a rich set of reagents that can move forward for development of diagnostics. So for example, you imagine you have an isogenic cell line pair, one that has a RAS mutation, the other does not. You can mix them, the mutant 1 to 1,000 with wild type cells, or 1 to 10,000, 1 to 100,000, 1 to 1 million, et cetera, and use them for a variety of diagnostic assays, be it with cells, with immunohistochemistry, for example, in the case, let's say, of a translocation, or with upon isolation of DNA and sequencing, and even use the reagent mixed for generation of cell-free DNA assays. And this is great because we can characterize and normalize and determine sensitivity for any given potential clinical assay. So it's really a wonderful use of the technology. We're broken down into 4 main groups that I've kind of mentioned already. One is what we call Horizon Genomics, mainly the group in the UK, as well as our recent acquisition of both Haplogen in Vienna, as well as the SAGE group, the Sigma Advanced Genetic Engineering group out of St. Louis, which just allows us to very rapidly generate cell lines through genomic engineering, be them diploid cell lines, or in the case of Haplogen, there's a Haplogen cell line that we use. It's almost like using yeast genetics, but with mammalian cells. And then with the SAGE group in St. Louis, we can use CRISPR-Cas9 to generate a variety of in vivo models. And I suggest, if interested, just to go to our website, and there's a tremendous amount of information available about these various initiatives. I've already discussed the reference standards that can come out of the use of the cell lines and mixing and matching mutant and wild type for a given loci. There's the bioproduction group, which essentially comes out of our gene engineering strategies so that we can optimize for cell-based models for production of, for example, biologics. And really what I want to discuss today was our research services. You know, we have a wealth of experience over a number of years with many examples of providing value to customers using siRNA, shRNA, and sgRNA for genetic screens for target ID, as well as target validation. We've also have a wealth of experience in terms of relatively complex disease models, in particular for oncology, be it 2D screening cells using tumor cells in 3D, mixed cells cultures, tumor microenvironment type assays that, again, going to our website will provide a wealth of information with respect to our activities. But what I want to do today was to give you a focus on our efforts here in Boston with respect to combination screening. Something, again, that we started to do about 12 years ago when we were at that point in time combinatorics. And so what I wanted to do was just, I guess, I think it's important at times to just state the obvious because it just highlights how important what we do is, which is to provide a few slides just to discuss why combination drugs are important for treatment of cancer, and then give you a little bit of an overview of our strategy for combination high throughput screening and what our mindset is in terms of how we approach this. And then, given the limited amount of time we have, I mean, I've done over about 70 projects, but given the limited amount of time that we have, I'm just going to really highlight one, which is around a project we did with Amgen on their MDM2 inhibitor discovery program that we worked up with them a couple of years ago. And then I wanted to spend a little bit of time, since it's such a robust platform, and we can identify often quite a few potentially interesting activities, combination activities, is just touch a little bit briefly on how we might prioritize combination drug leads. Spend a slide just talking about the fact that we can not only identify potentially interesting synergies, but we can identify very easily antagonisms when two drugs paired give rise to antagonism. And then I want to spend just 2 slides essentially on immuno-oncology, an effort that we're actively focusing on, and then give you a summary slide. So in terms of why we do combination screening, I mean, one reason it's come out from a lot of beautiful work from the folks that are contributing to Cancer Genome Atlas, that should be TCGA, not TGCA, as well as a lot of beautiful work from individual labs around the world where with the decrease in cost in terms of sequencing DNA, many groups have moved forward to characterize in detail tumor samples for various indications. And one thing that's quite striking and of course is rather depressing is the amount of complexity in terms of the type of dysregulation that's observed across the various tumor types that people have looked at. And so what you see here is work that was published in 2008 with respect to lung adenocarcinoma, either looking at the level of the various receptor tyrosine kinases shown here, or downstream through RAS or PI3K, that you can see at a reasonably high frequency, not only dysregulation, being it mutation, deletion, or amplification, but often you'll find that more than one event is occurring. And so what this has really kind of highlighted is, at least for me, is that, you know, given this, what we're encountering here in terms of this mutation burden or dysregulation burden, it's highly unlikely that one agent is going to be particularly efficacious unless a tumor cell is just completely addicted to that, for example, oncogenic driver. This is the case with respect to lung adenocarcinoma. I think I have one more slide here. With respect to brain cancer, essentially showing the same thing. This receptor tyrosine kinases, RAS and PI3K, signaling altered in 88% of the cases, and a breakdown of what in fact has been observed, characterizing tumor tissue from patients. And this is the type of dysregulation you see for P53, 87%, as well as cell cycle, in this case RB in particular, altered in 78% of the cases. So again, it just really kind of highlights that this is a multi-genic event, and again, single agents are probably not going to get us where we need to go. There's another issue to consider, and this is actually a build, so I will add this additional slide. It's in thinking about signal transduction pathways and the development of agents that we'd hope would dampen a particular signal pathway, transduction pathway that's important for proliferation or survival. So here's a cartoon, various receptor tyrosine kinases, and you've got two key pathways, the PI3K pathway and the RAS-RAF-MEK pathway. And what was identified a number of years ago, for example, that if you use a molecule that would inhibit mTOR, in this case the early work was done with rapalogs, what was found is quite surprising but very impressive how the tumor cells can adapt to the agents that you would develop to try to kill them, is that tumor cells that required mTOR signaling, you block mTOR signaling, you get an upregulation of AKT, a feedback upregulation of AKT to further stimulate this pathway, or you can have another pathway, in this case RAS-RAF-MEK. Particularly, you look at MEK, you see MEK increase in phosphorylation, which is evidence of increase in activity. And the wonderful thing about the work that gave rise to these conclusions is that we had reagents, for example, to track AKT phosphorylation or to track the RAS-RAF-MEK pathway activation. And so we would map out what the tumor cell would do when you treated it with a particular agent designed to kill it. The problem is that in the cell, signal transduction doesn't look like this, and in fact it looks like this, which I affectionately refer to as a fur ball as relates to signaling. It's very complex and it's very dynamic, where proteins talk to potentially many other proteins. And when you were to dampen a signal for any one given protein, we don't necessarily know how the tumor cell might try to compensate for that event, or what other redundant pathways might be operative. And I think particularly important, we don't necessarily have the reagents to be able to track what's going on when we use a chemical perturbagen to block signaling or survival pathways in the tumor cell. And so with that as the setup, having the limitation with respect to tracking what's going on, what combinatorics decided to do about 12 years ago was to, and it being important to use combinations to, in fact, ultimately be effective, was to set up a platform to do combination high-throughput screening. And what we've been interested in, and all the companies now are interested in doing this, is that they'll have a drug of interest. It's shown here in black. At increasing concentrations, there'll be an increase in effect. It can be effect on proliferation, it can be effect on survival. And what we're looking for here is the second agent that, when added to this first agent, will give a boost in effect or a boost in potency. There's two key issues here. What is that second agent? And secondarily, what concentration of the first agent and what concentration of the second agent will give rise to these sorts of activities? So combinatorics started about 12 years ago to solve this issue by generating what's called here the dose matrix. And you can consider these wells of a 384 or 1536 well plate, where what's done is to seed cells in the well, tumor cells in the well, and to screen one compound increase in concentration, as shown here, and a second compound increase in concentration, and then capture all the different concentrations and ratios of both agents. And what's shown here is growth inhibition, where what we've done is to include a T0 at the time of drug addition, so that at the end of the assay, in this case measuring ATP as a surrogate for effects on proliferation, we can start to, at a whole well level, tease out a cytostatic effect from a cytotoxic effect. And so what's shown here is a single agent dose response curve for a cytostatic agent that plateaus at a growth inhibition or GI level of 100. It's cytostatic, but you can see for the second agent, it's cytotoxic. And so what we do is we capture all the different concentrations and ratios of both of these agents. And then what we do is, with having good, if we have good single agent activities, what we can do is model essentially a null interaction, the equivalent of combining drug with self, and this is called the Louvet model. And so this is modeling here is based on the information with respect to the single agents here. Having this information, the null interaction, we can subtract that from the actual measured activity, excuse me, to get what's in excess. And so here's a heat map of excess over the null interaction at the various concentrations and ratios. And what's really great is if you have information about target relevant concentrations or have information about clinically relevant concentrations in the context of this type of screen where you see these sorts of activities, you can start to prioritize or deprioritize any of a number of the combination activities you might observe. And really a key point here is that we've spent over a decade to define and refine this process so that we can very, very quickly screen thousands of combinations to identify those where you'd see, excuse me, potential combination activity, and then move forward to evaluate them in more detail. Now, we have also, over the past decade, developed software that allows us to do this analysis. It's proprietary software that we can provide to other companies, and actually a couple companies have them installed globally throughout their organization. That includes not only the models for like Louvet or Bliss, but a variety of other models and that one can use interchangeably. And the idea is that once you have this data, you can evaluate a variety of ways. Now you get this excess surface in terms of Louvet excess. We can also boil this down to a score, any variety of different scores, but one that we use rather routinely is what's called the Synergies score. And it's a positively gated number that's positively affected by the concentrations where the effects are observed or the max effect for the combination. And so both of those will give you even higher synergy score, and that can be useful in terms of prioritization. But with this scoring system, we can then step back and look at combination activities from a global standpoint, where we'll have a matrix where we'll have the various cell lines that were screened and the various combinations that were screened. And when we screen a reasonable sized library, let's say it's 100 or 200 compounds, if we have mechanistic redundancy, one of the first things you can do is to look to see if other compounds that are in the library that hit the same target or pathway score. And so in the context of a primary screen, you can have validation with respect to combination activity. We can also cluster based on tumor type or specific types of dysregulation for tumor cells to see if combination activities track with a particular trait for the tumor cells that we're screening. I'll show you in this next slide here is that we've got a cell line collection from commercial sources that's at this point greater than 800. And that's nicely supplemented by an ever-increasing collection of isogenic cell line pairs for various tumor cell lines. And really, it's the importance of this core cell line collection cannot be understated. We started to collect the cell lines in earnest when we first started working with Novartis about 5 years ago. And the really wonderful thing there is that in parallel, Novartis started working with the Broad Institute on the generation of the Cancer Cell Line Encyclopedia. So we sourced the cell lines from the same sources used by the Broad Institute for their studies. And just as important, we used the same growth conditions as the Broad Institute had used for all their studies, in particular for generation of gene expression data. And these growth conditions can vary quite considerably across the cell line collection. It's not the same growth media for all the cell lines. And so what we have is really the best match to the CCLE of anyone outside of the Broad Institute and Novartis. And the data is available for our screening. We have about 90% overlap with the CCLE. The data for the CCLE is available when doing the data analysis for the information that we provide to the various companies. I think just as important, if some of the cell lines are not in this collection, or some of the genes that folks are interested in are not present in the CCLE database at this time, we're happy to provide cell pellets so that the folks that we work with can take those cell pellets and either do their own gene expression work or sequence genes. So I think finally, just as important, we can provide cells, for example, fixed. There are a number of companies that will want to look at cell surface expression, for example, if we're doing an antibody-drug conjugate screen. So this next slide just kind of provides an overview of kind of some of the things that we've done and some of the companies we've worked with. And it's a slide that I'm particularly proud of because we've worked with a lot of different companies, both big pharma, overall 11 of the top 14, as well as a lot of biotech companies. And I can provide the names shown here because this information has been made public. But for example, with Novartis, and Amgen, Abbott, Johnson & Johnson, Sanofi, Eisai, we've done multiple projects over multiple years. And that's important because it really, in our minds, helps to validate what we do in terms of the value and the fact that we're cost effective and really can deliver the data quickly. I think also what's really important is that with respect to many of the companies we work with, we have not only provide the work as a CRO, where I should highlight that the folks that we work with, they own the IP in its entirety, but separate and distinct from that, is working as a CRO with respect to Novartis and Amgen, for example, that we are co-authors. Folks here are co-authors on publications from Novartis as well as Amgen last year. We co-presented work with Johnson & Johnson at AACR in 2013, with Sanofi and Eisai at AACR in 2014, with Curis at AACR in 2015, and Infinity just a couple weeks ago at the ASCO meeting. So we're considered just as much collaborators, I think, as a CRO, which again is very important to us. We help in the design of the projects as well as in the data analysis and working out the next steps, as well as with publications and assisting with respect to IP filings. Now, we don't only do oncology. We've worked in a number of other therapeutic areas, including infectious disease, inflammation, in this case, Huntington's disease, spinal muscular atrophy, cystic fibrosis, and Duchenne's muscular dystrophy. And so we're quite versatile with respect to the type of assays that we can run. And I think particularly important is that we can do, for all these indications, we can do relatively large projects in a very quick period of time, delivering quality data. And a number of these companies now can do combination screening internally. They know what it costs. They see that we're cost effective with respect to our efforts. What I want to do in this slide is to just show you the two types of main screening efforts that we do for oncology. One is a pairwise screen where we'll take a collection of compounds and we'll screen them with each other across a number of cell lines. And this is the type of work that we have done with Novartis for a total of about 4 years. And it can, in this case, we had their entire pipeline, a number of approved drugs and a number of emerging drugs. And it really helped us in terms of defining and refining the process of doing this large type of screening. And this is a grid format. These are all potential synergies. They're given a synergy score, and the higher the score, the darker the color. So it just kind of points out the type of data that you may see. A more standard type of screening is the enhancer type of screening that folks work with us on, where they'll provide a candidate molecule. We'll screen it across a collection of compounds. It's usually 100 to 250 compounds. and we'll panel across a series of cell lines, anywhere from 10 to 100. And again, we can do this very, very quickly and generate quality data. We have a variety of different screening formats that we use. One, for example, is what we call the 6x6 format, as shown here. And the importance of the screening formats is that, well, two things. One is that for a number of projects that we've worked on, you don't necessarily need to go the route of a 6x6 format. Let me show you the example here, which is a 2x6, where you have a first agent that is active at increasing concentrations, but a second agent, there's no phenotypic effect as you go up in concentration, but you know you get target engagement at this top concentration. And so you might pick this strategy versus this one as an initial type of screen. But the other consideration is that given the sizes of some of the screens, it's more effective from a plate real estate perspective and a timing perspective to perhaps take a strategy like this, which is the 6x6 optimized, where it's essentially checkerboard, where we do every other measurement. And we've done a number of experiments here, both in silico, where we'll take data generated by 6x6 format and pull out every other data point, and then we'll do a correlation analysis to see how similar they are, and we know under what circumstances they'll be quite similar and when to go this route. Another one is the hybrid fixed dose ratio strategy, is shown here. I just want to show you an experiment in the next slide, where what we've done is this is the hybrid fixed dose ratio to orient you probably to start here, where you've got this full 6x6, and here's the excess that's observed, the Louvet excess, and here's now experimentally the hybrid fixed dose ratio, and to show that you can essentially pick that up quite fine. The synergy scores are quite similar, and it also holds true for even a combination effect that is quite weak. So we can go this route, and the importance again is that we can cast a wider net quickly and for a given price point. And so for some projects, we will not use this strategy here or a higher resolution dose matrix, but we'll go to an optimized 6x6 or a hybrid fixed dose ratio. So what I want to do in the next couple slides is just briefly review with you some of the work that we did with Amgen, who came to us a number of years ago with their MDM2 inhibitor. Clearly a disease-relevant target, a disease-relevant molecule, reactivate P53, be useful for P53 wild-type cancer cells, but clearly in and by itself is not going to be very effective as a monotherapy, in particular given that one of the genes that's most frequently mutated or inactivated in cancer is P53. And I should say that it's the case here with MDM2. I see it sort of as a stressed asset, even though it's clearly disease relevant. You could think of PI3K inhibitors as, to some extent, distressed assets where, except in limited circumstances in clinical trials, the PI3K inhibitors are not particularly effective as a monotherapy. An exception, of course, being the PI3K delta inhibitors for certain B-cell malignancies. So we see in the case of the MDM2 inhibitors and a variety of other molecules that folks are moving forward with, the writing's already on the wall in terms of the efficacy as single agents, how sustained the effects are as single agents. So they're coming to us to do these combination screens. And so the focus here was to take their MDM2 inhibitor and to screen it in combination with 1,200 potential enhancer molecules. This library included standard of care, a number of emerging drugs, some well-defined molecular probes, and given the size of the library, some somewhat ill-defined molecular probes. The idea was to screen in 10 cell lines, which I'll get to in a second. The matrix screening strategy was this optimized matrix that I've just discussed in the previous slides. And so we went ahead and did this screen with, as I said, 10 cell lines. We used 7 wild-type P53 cell lines where the MDM2 inhibitor is active, and 3 that were mutant. The idea being that if we saw activity in wild-type and mutant, it would be due to an off-target effect. And this is just a summary of some of the hits that came out of the screen. I should mention that I mentioned earlier, we published some of this work with Amgen, and that article can be found at the link here. But this is now a heat map to orient you. These are different compounds that scored positive in the screen in combination with the MDM2 inhibitor. And what you're looking at is the amount of synergy, and here's the targets that were identified. So here's a pure discovery project. We had no idea what to anticipate in terms of potential hits from this screen, pure discovery. And what you can see is that all these require the P53 be functional, because we're not observing any of these in mutant P53 cell lines. So here's the heat map, and so the strategy moving forward here after using the optimized matrix was to use a higher resolution dose matrix, the idea being that we wanted to more carefully examine the concentrations where you'd see these effects, so we could examine whether synergies were observed at target relevant concentrations and clinically relevant concentrations. So we took a number of the lead combinations and we screened in 40 additional cell lines, and in particular we're interested in a number of genetic backgrounds to pressure test to see if combination activity was increased with some of these genetic backgrounds, and I'll touch on that in a minute. But here's now this higher resolution dose matrix is shown here. This is a 9 by 9. What I've done is just to pull out in two different concentrations of the single agents and then the combination, just to show you there's a really nice effect. But again, we're casting a wide net and we're examining this at all the various concentrations and ratios of the two drugs. And so what you can see is here is the screen of the heat map, and the focus here was to not only do this higher resolution dose matrix, but to also, for a number of these leads, to examine the activity for compounds that had different scaffolds. So in the case of the MDM2 inhibitor, there's a molecule C15 and C25. In the case of MEK, trametinib, the PD compound, and then here are 3 different RAF inhibitors. The idea is to, number one, validate these activities and just make sure that they were due to on-target effects. So here's the heat map, and given that we're working with MEK inhibitors and RAF inhibitors, the question was, well, does it matter if you've got RAF or RAS mutant cells or if they're wild type? And the answer is no, that you see it whether they're mutant for RAS or RAF or whether they're wild type. But what still holds true is that you do not see the activity if the cells that we work with are mutant for P53. So we did the analysis shown here, and also given some of the leads that came out of the screen, PI3K inhibitors, MEK inhibitors, RAF inhibitors, we did a number of 3-way combinations as well. And I don't have time to go through the 3-way combinations, but some of that work is described in this link. And I think another thing really quite important to mention is that in this link to this publication, what the folks at Amgen did, they did a phenomenal job with this project, is they took a number of these combinations and they moved them forward into animal xenograft studies and showed that the activities that we identified with our in vitro screening platform recapitulated in an in vivo setting. And that's true for Amgen, it's true for many of the companies that we've worked with when we've identified these combination activities. That just gives you an overview of what we've done with respect to the Amgen project. Again, just one of over 70 that at least I've worked on in the number of years that we've been doing this as a CRO. And you identify a number of interesting potential combination activities. And really the question is, if you got a handful of them, how do you prioritize the ones that you should move forward with? And I've already kind of mentioned one way that you can do that, and that is that you're generating the dose matrix, you're screening the agents at a variety of different concentrations. You can look to see if the activities observed are happening at target relevant concentrations if you have that information. If they're approved drugs, you can do a preliminary filter based on whether you think it's in a clinically relevant concentration range, and then you can prioritize or deprioritize based on that. You can also, for example, if you're screening a large cell line panel, you can look to see if there's reasonable breadth of activity. But what I want to show here, and in more detail, this information and additional information provided in 2 publications that we did a couple of years ago, are some examples of how we prioritize some of the activities that we've identified. One that I've kind of touched on already with Amgen is just do a xenograft assay to see if you see the combination effect. So what's shown here is percent survival for a particular combination we worked on for vehicle, the 2 single agents, and then the combination. Another type of assay that we've worked with to prioritize or deprioritize combinations is shown here. This is work that we did with folks at the Dana-Farber Cancer Research Center, Doug McMillan and Constantine Mitsiades in Ken Anderson's lab, where we essentially took cells from patients that had multiple myeloma, put them in a 3 to 4 well dish, and then screened with our 2 agents of interest, where we saw synergy with multiple myeloma cell lines, and we saw with a very high frequency synergy with cells from patients, which was very exciting. We've also done work where we've screened tumor cells in culture with, in this case, bone marrow stromal cells, as being multiple myeloma, where we can screen for the effect of the combinations with the tumor cells alone or in combination with the stromal cells in a mixed culture, or taking a variety of factors that would be present in that bone marrow microenvironment. For example, IL-6, do the assay minus and plus IL-6. And this, again, this is not growth inhibition, but it's inhibition. This is inhibition, and this is the excess activity. Again, an indicator of synergy. But again, these are various ways that you can prioritize or deprioritize a given combination activity. And then, as I mentioned at the beginning, in addition to the ability to identify strong synergies, and this is that hybrid fixed dose ratio that I was talking about earlier, where you're seeing these nice synergies within these 2 cell lines, in this case with an antimetabolite and an apoptosis inducer, or a particular kinase inhibitor and an apoptosis inducer. We can just as readily identify instances where you have antagonism. So in this case, it's a DNA damage inducer and a compound that induces mitotic arrest. You have lower activity here than you have with respect to the single agents. And this is just a cluster analysis for these 2 groupings of agents, just to show that you can nicely cluster these across a variety of cell lines as shown here. So it's incredibly powerful for both identification of synergies as well as identification of potential antagonisms. Now, I kind of wanted to end with that slide with respect to target agents, but just really in this slide make the following point, which is that, a couple of decades ago when folks started to develop chemotherapeutic drugs, what became obvious early on was that there was better efficacy by use of drug combinations. And the strategy at that point in time was if you had an agent or a drug that had activity as a single agent in the clinic, it would be paired with everything else that would have activity in the clinic. The other strategy was to pair drugs based on what you thought you knew about the disease biology. And that's held true with respect to chemotherapeutic agents and now over the past decade or so with respect to target agents as well. It's really been sort of a limited pairing of agents based on the fact that they have some single agent activity, or we have a hypothesis based on the disease biology. And I think what's really been transformative is to have this ability to do combination high throughput screening, to now screen in combination drugs to score for potentially beneficial effects by use of drugs in combination. And what I really didn't highlight earlier, but I think is particularly important, is that the ability to do this in large scale is incredibly valuable. To step back and not make any real strong assumptions up front, but just do the experiment, to be agnostic, to have that collection of approved and emerging drugs and well-defined molecular probes, and to have a really large cell line panel with good genetic diversity. It's very rich for discovery, where you very frequently obtain some very interesting leads, and really the key is to have those secondary filters in place to convince oneself that what you have is disease relevant with the potential for translation. That's how chemotherapeutic drugs as well as target drugs have tracked, and I think what's become quite obvious, at least to me, over the past couple of years, tracking what's happening at the various conferences and in the literature, is the same is going to hold true with respect to immunomodulatory drugs. We're going to need to figure out how to use them in combination. And I think just as important, since so many of them function through cell signaling, them being receptors, that there's probably going to be significant benefit by pairing them with targeted agents. And again, the importance of being agnostic with respect to the screening. And having said that, I just want to touch on a slide here, which is kind of old work on our part here at the Horizon Discovery Boston group, is that we've used this platform extensively for identification of agents that would synergistically block, in this case, the production of pro-inflammatory cytokines. And so this is a T-cell receptor activation assay with immune cells from patients purified from PBMCs. What we're measuring in this case is TNF-alpha production, 2 different drugs, drug 1, drug 2. Again, this excess activity that's observed, which is a measure, a way to identify the synergy. And then what I've done is to pull off, to graph the single agent activity for drug 1 with either 1 concentration or a 2nd concentration of drug 2 to show that you can see these nice potency shifts and efficacy boosts. And so we're taking this strategy and moving it forward with, I think, considerably more complex immune cell assays to examine how immunomodulatory drugs will behave in combination. And so this is my summary. With respect to combination drug discovery, we have over a decade of experience with both oncology and with respect to combination high-throughput screening. What I really didn't touch on is, when we perform these screens, we often get very interesting activities, and we can sometimes do the validation based on a chemical genetic strategy, based on the mechanistic redundancy we have in the library. But it's nicely complemented by our SH, SI, and sgRNA technologies that we have also at Horizon Discovery. Showing you the list, a small list of companies that we work with, should be quite clear that we have a strong track record with respect to technology development. A really key point here is that we have a proven ability to very quickly deliver actionable insights that can help move programs forward with the hope that we'll have a very strong impact as well in the clinic. The assays need to be robust in case of known immunomodulatory agents, but also with respect to our standard oncology assays. We have 3D4 and 1536-well assays. We need to be able to capture the disease complexity, and we've worked hard in terms of doing that, if not in a primary screen, so that because we need to be more robust by clever use of secondary assays. And I think really the bottom line here is that the combination drug discovery is a critical path for cancer drug development, and involves both the synergistic selective killing of tumor cells and the identification of synergistic immunomodulatory drugs. And we're nicely poised and actively to do that, and we're actively engaged in delivering significant value to the folks that we're working with. So that in a nutshell is really what I want to cover today.